Hi, I'm Andy Burrows, here for Tides TV. I'm here with Dr. Marie Wikström Lindholm, who's VP, Head of Technology Innovation at Silence Therapeutics. Thank you very much for joining us today. So you're speaking here at Tides on GalNAc conjugates. Some people would say there isn't anything left to investigate. I assume you don't agree with that?
Yeah, no, definitely. So I came here being ready to make some statement about this because it's been very clear at meetings over the last couple of years that although everyone has confirmed now that the GalNAc conjugated oligonucleotides, may it be single or double-stranded, really works. I mean, that's basically revolutionized our industry because it makes it possible for us to get really good delivery to the hepatocytes, so we can reduce the dose, we can do something about some tox effects, etc., etc. So all that is really positive. And in my daily life, I frequently meet people asking, why are you working with the GalNAcs? Because that is already shown to be a really good idea. Is there anything left to do? So my presentation later today is going to be describing some of the things that we have done in-house at Silence, improving this, because we think that there is still plenty of room for improvement. And because of that, and because this is something I feel quite strongly about, it's actually been a pleasure to attend several of the talks earlier during the meeting, because it seems as if quietly everyone else in the industry is thinking the same thing. We have something that works, but it doesn't mean that we can't make it better.
Yeah. And so where do you see the biggest areas of potential there still are?
Well, as I said, we know that it works, and that's all really good. However, if you look at the data showing how much of the injected dose, because these compounds has to be injected sub-Q,
right,
then first of all, we lose about 80% of it within the first 24 hours. It goes out in the urine. Everyone knows that. And then it's actually just a tiny fraction of the remaining compound that reaches the final destination. So one of the things where we really do have room for improvement is to try to increase, because we just heard that our compounds are very expensive. They're going to continue to be very expensive. And if you can increase the amount that actually reaches the final destination, that would be the cytosol or nucleus of the hepatocytes, by just a factor 10, that would be a huge advancement, because it would make it possible to reduce the dose, and there really is plenty of room for improvement here.
So hopefully lots of areas to improve. What challenges still need to be overcome to reach that point?
Well, I can talk about this probably for a full day, but I think it all boils down to, we know that just a tiny fraction reaches, as I said, the cytosol or the nucleus, depending on mode of action. We know that it gets into the hepatocyte, and we know, looking at all kinds of different images, etc., that it's just less than a promille that actually gets out. In order to get to the right point, it needs to do what's called endosomal escape, and that sounds easy, but it's been a burial ground for so many efforts in the past, because we wanted to get into the cell through a pathway that the cell has used millions of years to evolve a defense against, because basically we want to do the same thing, getting foreign genetic material into the cell that cells had evolved protection agents against, because that's the way that we get infected by virus and bacteria.
Right, okay, yes, interesting stuff. And so what next for you and Silence?
So what's next for us in Silence is to try to do something about this in our own way. So we have now been working under the banner less is more. So we have quite complicated molecules, all of us do. We have certain modification patterns, we have what's called the GalNAc tricluster, and we have been, in a quite systematic manner, been looking at, for that delivery method, to see how much of that is really necessary, how much can we simplify the molecules, and by doing so, because that was the starting point, we have actually found several really interesting made some interesting discoveries where we saw that not only could we simplify the molecules and keep activity, we were actually able to improve it. Okay. So that's one thing that we are doing. And also, just to be clear, I'm not going to come here with the message that we don't care about getting into all other cell types. Of course we do. And extrahepatic targeting is also very, very important. But we do think that, first of all, the GalNAc conjugated oligos can get better. We also think, of course, that some of the basic structural features that we now are using in the new and improved GalNAc conjugates could be used for other targeting moieties as well.
Right. Okay. Well, good luck with everything, and thank you very much for joining us today.
Thank you.